Hi everyone, welcome back to the third episode of this immunology series. In this episode, we'll be drilling more into the B-cell activation and the antibody response, and we'll be covering T-cell independent activation, Th2 activation, and T follicular helper cell activation. So in the last episode, we briefly covered how Th1 cells can activate B cells, but actually all sorts of T helper cells can activate B cells. And a cool thing too is that B cells can even activate themselves. And this depends on the type of the antigen in the environment and also the composition of cytokines. So because this is one of the initial responses to infection, the main immunoglobulin responding is IgM. And that is the main immunoglobulin driving this T-cell independent response. For the first signal, the B-cell receptor identifies antigens in the environment such as bacterial polysaccharides or lipopolysaccharides. The second signal also arrives independent of T-cells, and that can either involve the complement system or toll-like receptors that identify pathogen-associated molecular patterns, also known as PAMPs. So this T-cell independent B-cell response generally happens in the extrafollicular space of secondary lymphoid tissues. So after activation, these B cells can turn into short-lived plasma cells, also known as plasmablasts, and they secrete IgM. In comparison, this T-cell independent response can also develop long-lived plasma cells, although it is more common for plasma cells to develop in the germinal centers of secondary lymphoid tissues, because that's where they're able to undergo maturation and develop a higher affinity for antigens. These long-lived plasma cells play a much larger role in the later stages of infection. However, this T-cell independent response might limit the ability of class switching, and the production of memory B cells might be limited. Now we'll dive into how Th2 cells are important in the B cell response. So let's assume that this Th2 cell has already been activated by a dendritic cell in the lymphoid tissues. Now it's ready to respond and recognize this antigen that's being presented by a B cell. So the B cell is presenting the same antigen that the Th2 was previously introduced to. The Th2 cell recognizes it, and they go through the three signals required for activation of the B cell. The first signal involves antigen recognition and binding. The second signal is the T cell and B cell interactions involving the CD40 and CD40L ligands. And the third signal involves the cytokine instructions, which help to guide the B cell to mature into a plasma cell or memory B cell. The composition of cytokines in the microenvironment help to determine the class switching of immunoglobulins. IL-4 also induces the class switching of B cells into IgE secreting plasma cells. IgE antibodies defend against worms and parasites through mast cell and basophil activation. But for the same reasons, the Th2 response can exacerbate asthma, allergic diseases, and autoimmune reactions. B-cell maturation by Th2 cells also results in the production of memory B cells, and these cells are helpful in the secondary phase of infection. When the B cell matures into a plasma cell, the plasma cell is no longer capable of class switching because it no longer displays an MHC2 on its cell surface. It also has very few B cell receptors left because most of them are being secreted as antibodies. But the few B cell receptors that it retains are really important because when they detect antigens lingering in the environment, it helps to prolong the lifespan of the plasma cell. Now that we've covered the T cell independent and the Th2 activation of B cells, we can cover the T follicular helper cell response. So it helps to really visualize where all of this is happening. So here is a lymph node as an example. T follicular helper cells are also called Tfh cells, and they are really important in these secondary lymphoid tissues because they help to create these germinal centers, and that is where they help the B cells mature and develop higher affinity and class switching. So first we'll cover the 4 steps of Tfh activation. Step 1 involves this migratory dendritic cell. that enters the lymph node and either presents the antigen to a resident dendritic cell or presents the antigen to a naive T cell itself. This first step occurs in what's called the T cell zone. Step 2 involves the dendritic cell moving towards the T cell B cell border. Step 3 involves the commitment phase. When antigen concentrations are high enough, the B cell actually becomes the antigen-presenting cell here. The B cell then presents the antigen and other signals so that the TFH cell can fully differentiate. Step 4 is called the effector phase because this is where the TFH cell can demonstrate its effector functions. They help to create the germinal center where it can promote the survival, expansion, and affinity maturation of B cells. In the primary B cell response to an infection, the TFH cell enters the germinal center and begins interacting with B cells, and that helps the B cell to mature into plasma cells or memory B cells. These B cells can mature into short-lived plasma cells that can leave the follicle and play a large role in the initial stages of infection by secreting these IgM antibodies. The B cell can also mature into a long-lived plasma cell that plays a larger role in the later stages of an infection, or the B cell can mature into a memory B cell that stays in the germinal center where it continues to undergo selection. After B cells in the germinal center become activated, they can become memory B cells, and these play a large role in the secondary infection. If there is a reintroduction to an antigen, memory B cells can quickly differentiate into plasma cells capable of secreting up to 3,000 antibodies per second. If the memory B cell doesn't mature into a plasma cell, it stays in the germinal center, where it undergoes further selection and maturation while it's awaiting the next round of infection. So that's it for the 3 main B cell responses. If you've enjoyed this episode and would like to follow along, you're welcome to follow my channel here, or you can also find me on patreon.com, where I'll be posting more exclusive content and offer some other fun areas of collaboration. I have a couple more videos in this immunology series, and after that, I'll be posting a lot more content on the environment, on food manufacturing and policies, and just health and wellness in general. Hope you enjoyed learning, and I look forward to seeing you on the next one.